Today's AQA required practical is on enzymes. You will investigate the effect of pH on the rate of reaction of the enzyme amylase. For this practical, you will need the following equipment and solutions. You will need 1% starch solution, 1% amylase, 1% iodine, a range of buffer solutions. Here are pH 3, pH 5, pH 7, pH 9 and pH 10. You'll also need a spotting tile, 3 measuring cylinders, 10 centimeter cube, a stopwatch, a stirring rod, test tube rack and 3 test tubes, and you will need a water bath. Here I have an electric one, but you can use a beaker with hot water and a thermometer which can act as a water bath. First thing I'll do is put my goggles on. I'm going to demonstrate using the buffer solution pH 5. I've labelled my test tubes A for amylase, S for starch and B for buffer. So I'm going to put 2 centimeter cubes of amylase into my first test tube. Using a different measuring cylinder, I'm going to put in 2 centimeter cubes of starch solution into my second test tube. I'm using a different measuring cylinder. I'm going to measure out 2 centimeter cubes of buffer solution pH 5. Which will go into my third test tube labelled B. I will then put my test tubes into the water bath, which has a temperature of 30 degrees Celsius, and I'll leave that there for 10 minutes. Using my spotting tile, I will put 2 drops of iodine into each of the dimples. After 10 minutes, I will then put the amylase solution and the starch solution and the buffer pH 5 solution into one test tube. Give it a mix with a stirring rod. Go back into the water bath using a pipette. Take the first sample, put it into my first dimple in the spotting tile, and you can see it is blue-black, which means that starch is still present. Then using my stopwatch, I'm going to time every 20 seconds and take a sample from the test tube and put it into my next dimple. You can see the colour changing already. It's turning a little bit more lighter, which means that the amylase is reacting with the starch and the starch is being digested. Slight change colour in the third one. And there's no colour change in the fourth one, which means that by 80 seconds the starch has been digested by the amylase. What I'm going to do now is repeat the experiment using different pH buffers, such as pH 3, pH 7, 9 and 10. Once you have completed your practical, you can plot a graph like this. You can do time it takes for the amylase to completely break down the starch against the pH of the solution, and the graph should look something like this, with the optimum reaction taking place between pH 5 and 7.